Our speaker tonight is David Inouye, and he is retired from teaching ecology and conservation biology at the University of Maryland. But for many years, he's conducted research at the Rocky Mountain Biological Laboratory in in Gothic, Colorado. So he is long familiar with Colorado. He's been working here on research projects for the Rocky Mountain Biological Laboratory since 1971. And tonight he's going to be talking to us about some long-term studies that he's been working on. And long-term studies are just kind of what I call the gold standard of research in biology. We just don't get a lot of those projects, and they're much more meaningful in the long run, particularly for issues like conservation. So I'm looking forward to tonight's program. David has served on those long-term projects, including banding hummingbirds, studies of plant demography, individually tagged plants followed for up to 52 years, and he studied the phenology and abundance of flowering plants flowering by about 125 species of plants. That study is now in its second generation of researchers. His son and daughter-in-law have picked up that project and are continuing it. This reminds me a little bit about the research team in the Galapagos who have done those long-term research projects down there uh, for a couple generations. He spends his winters in near Peonia. He serves on the boards of Citizens for a Healthy Community, the North American Pollinator Protection Campaign, the Endangered Species Coalition, and Hummingbird Conservation Networks. And I am looking forward to your talk tonight, David, and I'm going to turn it over to you now.
Thank you. And let's see. Okay, is that title slide showing up all right?
It is.
Okay, thank you. I'd like to acknowledge the Rocky Mountain Biological Laboratory, which has provided the venue where I've carried out these long-term studies, and thank those of you who pay taxes, because some of a very small proportion of your tax dollars goes to the National Science Foundation, which has helped support this long-term research and generate the data that I'll talk about tonight. That hummingbird on the title slide is a male broad-tailed hummingbird, and I want to point out two things about it. One is that there's a yellow dusting on the chin of that hummingbird under its bill, and that's pollen from flowers that it's visited, so it's a good pollinator. And the other thing is that many of you have probably had the opportunity to hear the whistling noise that a male broadtail makes when it's flying by, and that's made by this little slot between the first two primary feathers on the wing. So it's a mechanical noise that it makes as it's flying. And my first graduate student did a project where she cut about 2 millimeters off the tip of that first feather, and that was enough to silence the whistle. And so we could do experiments on what's the impact of losing your whistle. The study area that I'll talk about is in the West Elk Mountains. Those of you that are skiers may know the Crested Butte ski area. There's Crested Butte Mountain, but where I work is further up the East River Valley on the backside of Gothic Mountain, shown here in this aerial photograph. Normally, there's a lot of snow up at this altitude. The average since 1975 is a little over 10 meters of snow falling, with a range between 4.7 and 16 meters of snow. This year, as of yesterday, there's been 1.84 meters of snow, which is almost 60% below average. Probably by today, it's at 60% below average. So if we don't get some good snowstorms in the coming couple of months, we're going to be in a world of trouble. During the summer, this is what the view looks like. The biological lab is in the East River Valley at the confluence of Copper Creek, which comes out of this drainage in the middle, and that heads over towards Aspen. And Gothic was an old mining town. It was very busy in the 1880s for a few years while it was a silver mining boom town, but it became pretty much a ghost town and ended up being purchased in the 1920s by the founder of the Rocky Mount Biological Laboratory. And we're going to be celebrating our 100th anniversary in a couple of years. The arrow here shows my cabin, which is where I've been doing a lot of banding. I'm going to talk a little bit about climate because it's what drives a lot of what I talk about this evening. The global climate is creating regional climates, things like the cycle of the North Pacific Oscillation, which is a 50 to 75 year cycle, the El Nino Southern Oscillation, which goes back and forth between El Nino and La Nina, and that's a 2 to 7 year cycle. And then there's another one called the quasi-biennial cycle, a little over 2 years. And those cycles all interact to create our local climate. And the aspects of the local climate that I'm particularly interested in are snowpack, the date at which that snow melts, and then how much of it is rainfall, also temperature. And that local climate interacts with the regional climate to influence the phenology of migratory species like the broad-tailed hummingbirds, which are now overwintering in Mexico and then are flying back up to Colorado for the breeding season. It also influences the phenology of emergence of species that overwinter in place. Things like bumblebee queens, like this bumblebee queen visiting the larkspur flower. Also the phenology of emergence of hibernating animals like marmots and ground squirrels and chipmunks. And then the climate is also influencing the phenology of all the flowering that happens during the season and affects all the floral resources that are necessary for pollinating species. In 1973, a group of us, most of us were graduate students at the time, were interested in looking at floral resources. Some of us were studying hummingbirds, some of us bumblebees, and we decided one snowy day in June in 1973 that we'd collaborate. Each of us picked a different habitat and set up some 2 by 2 meter plots. And then 3 times a week, we would go around and count all the flowers of any species in those plots. Here's an example of one of those plots in our wet meadow habitat. There are a few flowers of the spring beauty already in bloom here. There are some buds of the Veratrum californicum or Veratrum tenuifolium, the false skunk cabbage or corn lily coming up here in the background. There had been an avalanche that winter, and so you can see some of the avalanche debris in the background, but fortunately it missed the plot. So we have 34 of those plots that we go around to every other day for the 3 times a week. And this is kind of a phenological calendar showing the progression of flowers that we count there. So starting with here up at the top is spring beauty, Claytonia. That's the first thing to flower after the snow melts. And then the willow shrubs come into bloom, the glacier lilies, the early delphinium, strawberries, violets, some of the Rigeron daisies that come into bloom, Oregon grape, and then monument plant or Frasera speciosa, roses, later sunflowers like the aspen sunflower, things like the Mertensia fusiform or Mertensia ciliata, the bluebells, and we get some of the carrot family umbels coming into bloom. Calochortus or the Mariposa lilies. And later in the summer, we start to get some Indian paintbrush, more late flowering species in the daisy family, and then ending up with some of the gentians. So that's the progression of different species of wildflowers that the bumblebees, butterflies, and hummingbirds are having to visit. I mentioned the glacier lily is one of the first things to come into bloom after the snow melts, and that would be the first resource that the hummingbirds, the migratory hummingbirds, the broad-tailed hummingbirds, have to visit when they first get there. There actually are some interesting color morphs. You can see the yellow anthers on this particular individual, but there are also some red anthers on some other individuals. And there have been studies that have used that natural color variation as a way of tracking the flow of pollen from one plant to another. It's a very common wildflower at the altitude where I work, and some of the meadows are quite yellow with all of the glacier lilies. And hummingbirds, although they visit these flowers and collect the nectar, probably do little, if any, pollinating. And that's because of a morphological mismatch between the hummingbirds. You can see this bird's bill poking into the flower, but it's not coming into contact with the stigma where pollen would have to be deposited in order to pollinate the flowers. So the almost all of the pollination that takes place is a consequence of visits by bumblebees. And so here's a bumblebee queen, and you can see that she's got a lot of pollen on her underside, both picking it up and dropping it off as she collects nectar. Oops. So Billy Barr is a colleague and friend at the Rocky Mountain Biological Lab who's lived there pretty much year-round since about 1974. And he started collecting information since he's there year-round about a variety of climate data and also some biological data, including when did he first hear one of the arriving male broad-tailed hummingbirds in the spring. So his data show that the birds are arriving now a few weeks earlier than they did when he started collecting data. And if we plot his data, in this case, I'm plotting the arrival dates against how much snow is still on the ground on the 30th of April. The red dots are the arrival dates of the hummingbirds, and the yellow dots are the first flower dates for the glacier lilies. And you can see that they both vary depending on how much snow is left on the ground, or I could have also used the last date of snow. They both show these kinds of relationships. But the slopes of these two lines are not parallel. And that means that these two different species are not responding the same way in terms of their phenology with regard to snowpack and snowmelt date. What this also means is that there's the potential, as snowpack is declining over the years, for the snow to be melting now well before the hummingbirds get there, and for the flowering to start before the hummingbirds get there. So that's something that we're continuing to track as the environment changes. The second species that comes into bloom at this site is the early larkspur, Nelson's or Nuttall's larkspur. And here's a male broad-tailed hummingbird visiting one of those flowers. They probably are doing some pollinating, but these flowers are also visited by the queen bumblebees, and they're also doing some pollinating. This plant gets to be quite common. This is a site at the Crested Butte Stables where they rent horses. And if the horses are not there too early, you can see that these wildflowers are very densely populated there. In another site at the biological lab, since 1991, I've kept track of how many flowering plants are there in a permanent plot. And you can see that varies a lot from year to year in terms of how many flowering plants are in this plot or in these plots. Here's a queen bumblebee approaching one of the flowers. And in this case, I'm showing the numbers of flowers from year to year, not just the number of plants. And the numbers of flowers also varies a lot from year to year. And I think I understand why, or at least a little bit about why. If you plot the numbers of flowers per stalk against how much snow fell in Gothic the previous winter, there's a pretty strong correlation. So the more snow we get, the more flowering plants there are, both in terms of the numbers of plants that come up and also in terms of the numbers of flowers that each of those plants has on its stalk. I haven't been able to figure out the causation behind this correlation. So I don't know why this is happening. I've tried some experiments like watering to see if it was related to soil moisture, and it turns out it's not. But there's something about the environment that those plants are experiencing underground during the winter that is driving that correlation between how much snow we get and how many flowers they make. The average number of flowers per inflorescence is declining over years. And that's a little bit worrisome because that means that there are fewer flowers and less nectar for the hummingbirds and also for the bumblebee queens to be collecting. Another thing that's changing is the number of aborted buds. And so here's a flower stalk of that larkspur. The flower buds are still pretty early. It's going to take them another week or so to expand to the point where they can open. But this is an aborted bud at the top of the flower stalk. And it turns out that there are quite a few of the flower stalks have those aborted buds. And the percentage of buds that are aborted can be pretty high. As many as 40 to 60% of the buds can abort. And the more snow there is, and the later the snow melts, the lower the percentage of aborted buds. And that is also a worry because snow melt dates are getting earlier, as I'll come back to later on, as we're getting less snow in the winter. And that means that there's a higher proportion of these buds are aborting and therefore not making nectar for the pollinators. The later species of larkspur, Delphinium barbii, has more flowers per stalk. It's also visited heavily by hummingbirds, in this case, a female broad-tailed hummingbird, and by bumblebees, in this case, worker bumblebees. And I think it's interesting to think about the bumblebees on their tongues and the hummingbirds on their bills and their tongues are carrying various yeasts and bacteria that they pick up from the nectar and transport from one flower to the next. And because these flowers can be visited both by hummingbirds and by bumblebees, there's undoubtedly some mixing of the nectar microbiome of fungus and bacteria that are being transported back and forth. But nobody has actually studied that yet. I've been banding hummingbirds for over 50 years at this site. And our technique now for catching the hummingbirds or banding them is to use a trap like this, which has a net side that you can pull up to give access to the birds. This is my granddaughter who's about to help me get this Rufous male hummingbird out of the trap, so we can look and see whether it's already banded and banded if it's not banded. In addition to the broadtails, we get Rufous, the Celosporus rufus, coming through as post-breeding migrants starting in early July. And starting about mid-July, we'll also get Calliope hummingbirds. Calliopes are the smallest species of bird in North America. And on a good day, I can catch all three species at once. In fact, here's not a very good picture of the hummingbirds, but here's a male calliope, a male rufous, and a male broadtail that all ended up in the trap at the same time. At the lower altitude where I have my winter home in the North Fork Valley of the Gunnison near Paonia, we have resident black-chinned hummingbirds. And actually, despite that name, the lower part of that gorget is a really nice purple, which grades into a black color at the upper end of the gorget. In not too far from Gothic, several years ago, we had a bird watcher who had an albino, apparently broad-tailed hummingbird, visit his feeder, and this is a picture that he took. So I'm unfortunately wasn't able to band that bird, but maybe it'll come back again someday. When we banned hummingbirds, first of all, we have to have federal permits, which are somewhat hard to get. You have to apprentice for a few years as a hummingbird bander. You also have to take a workshop in banding hummingbirds. And when you're approved and you're sent bands, they send you these aluminum sheets of bands. And one of the skills that you have to learn to become a hummingbird bander is how to cut up those bands and make them the right size for the birds that you're banding. And so this is a male broad-tailed hummingbird in my hand. At the same time, I'm holding some of those bands. And that was a picture from National Geographic magazine a couple of years ago. Up close, this is what the band looks like. It's a letter and 5 numbers and wrapped around the leg of the bird. In addition to the federal permits that you need, you also have to have state permits to be a bander. And I think in the whole state of Colorado, there are only 4 or 5 people who are banding hummingbirds. So unfortunately, we very rarely see birds that others have banded. But I do see hundreds of the same bird from year to year, same birds that I have banded, because they tend to come back to the same area where they were born. The oldest known hummingbird in the wild is a female broad-tailed hummingbird that we caught in Gothic back in the 1980s. We saw her almost every year for 12 years. So chances are she was maybe even 13 years old, and that's very old for a hummingbird. The oldest birds that I've caught recently have been 9 years old. I've caught a couple of 9-year-old females. The females live longer than the males. Average female is probably more like 4 to 5 years, and average male maybe more like 3 to 4 years. Among the flowers that they visit in our area during the summer are the paintbrush, Castanea. The little bump here at the end of the stigma is where the pollen has to be deposited, and the orange that you can see here inside part of the flower would be the pollen that would be deposited on the hummingbird's head. So hummingbirds are pretty good pollinators of that species. The shooting star columbine, Aquilegia elegantula, is another species that's visited commonly by hummingbirds. You can see a little bit of yellow on the underside of this female's chin, but the males also visit that. And because we've been looking at both the timing of flowering and the abundance of flowering for over 50 years now, we know that both of these metrics of floral resources, when they come into bloom and how many there are changing divergently for both the flower species and for the pollinator species. They're changing differently for the bumblebees and for the hummingbirds and for the butterflies. Another potentially important resource for females in particular is access to stream insects, because we think that a lot of what they're feeding their babies is insects that they're catching from those streams, things like midges that are emerging from the streams. And if you're have good eyesight, you might have spotted the hummingbird nest in this picture, which is right there. Female broadtail sitting on her nest. And unfortunately, that nest was preyed upon by a crow later, not maybe a week or so after I took this picture. And so no babies were produced by that nest. I'm collaborating with a stream biologist who works at the biological lab. And he says that these stream insects, you can detect they produce lipids, fatty acids, that are different from the fatty acids of terrestrial insects. And so we're hoping to demonstrate or document how much of the fat that a female hummingbird is putting on, or that her babies are putting on, is coming from the aquatic insects as opposed to terrestrial insects, but we don't have an answer for that yet. The area where we're working here in Southwest Colorado is close to one of the temperature hotspots for climate change, for temperature change, as you can see in this map. And so one of the things that I've been working on the last year or two is an analysis of the Crested Butte weather station data. That weather station started collecting data in 1910 and is still ongoing. And so I've been looking at variables like how many rainy days are there between May and September, and is that changing? And it has been changing. There's a curvilinear relationship that you can see that it was increasing up until the 1970s, 1980s, maybe even 1990s, but then it's been declining. And so one of the things I've been trying to do is fitting different statistics to these data points. And so you could have a linear relationship, which shows there's been a linear decline since the mid 1980s in how many rainy days there are, or a curvilinear relationship that fits the long-term data, the whole data set. Another example of something I've looked at is how many consecutive days are there with no rain in Crested Butte? And that's also fit by a curvilinear relationship. And it looks like maybe that metric is increasing, so that we're tending to get more consecutive days with no rain in Crested Butte than we had back from the 1960s up until maybe around the early 2000s. Another example, some of the climate models predict that variability is going to increase in climate variables as the climate is changing. And so I tried looking at the coefficient of variation, which is one metric of variability, in how much rain there is per rainy day in Crested Butte. And that seems to be increasing over time. All of these things I've looked at, you can see there's a lot of scatter. We can fit a line to those points, but there's a lot of scatter. And that's pretty common for climate data. And that's one reason you really have to have many decades of data before you can begin to pick out trends, not only for the weather data, but also for the biological variables that I've been talking about. Closer to home, these are weather station data from Gothic, which Billy Barr has been collecting since the mid 1970s. One of the things he's kept track of on a permanent spot near his cabin is what's the last day of snow, what's the first day to bare ground. And again, a lot of scatter. It's varied from the 23rd of April in, I think, 2012 to the 19th of June in 1995 or 1996. And that happened to be a winter that I was spending in Gothic, and the road didn't open until about mid-June that year. But the overall trend here is that the snow is melting earlier. And that's for two reasons. One is that we're getting less snow, and the other is that the spring temperatures are warmer than they used to be. The green bars here are the data that Billy Barr collected since the mid-70s. And I was able to use his data together with data from a gauge, a river gauge, water level gauge down in Almont, Colorado on the East River to hindcast or predict what was the snow like in Gothic back to the 1930s when that river gauge was put in. And the overall trend here is that there was a bit of a decline in the 50s and 60s, an increase until about the 1990s, and then a pretty steep decline since the 90s. So I can pull that trend out of this variability that I've been able to document. So that's not good for the ski area. It's also not good for water users that depend on snow and snow melt throughout the summer for irrigation, for recreation. So we seem to be on an unhappy trend here. Let me talk a little bit about some of the other hummingbird research that's going on at the biological lab. Here's a male broadtail, and you can see he's perched on the feeder at my cabin. He's wearing a band that I'd put on earlier, but this one's also wearing a pit tag, the kind of tag that you might put on that many pets have under their skin. So when this hummingbird flies through an antenna on its way to the feeder, we can tell which individual that was and when it came to the feeder. I've also helped to test a prototype of a kind of hummingbird feeder that has a camera, and it takes pictures of the birds that are sitting on it at the feeder, and I can tell from that camera that the bird is banded. I can't read the band, and it's using artificial intelligence to try and identify the bird. And unfortunately, when it made that attempt at identification a couple of years ago, it came up with a wrong name. That's Crested Butte background, mountain in the background you can see there. I also had the feeder up at my house in Paonia at one point, and you can see Bullock's orioles are also visiting the hummingbird feeder at the lower altitude. A researcher who was a graduate student at the time, now has his PhD, was looking at a couple of different aspects about hummingbirds at the Rocky Mount Biological Lab. He collected blood samples from a few hundred hummingbirds and used those blood samples to genotype those hummingbirds, and then was able to look at the relationship between their genomes, their bill length, when they arrived, and what flowers they visit. And so here, for example, is some date showing the date of arrival, or at least when we first caught the birds, depending on what their genomes were. And there's sort of two peaks here, an early peak and a later peak. And then he compared what were the bills measured either as the exposed culmen or the full gape of early arriving birds and later arriving birds, and found that there's seems to be a slight difference there. And so we're repeating that project now to see if we can duplicate that finding that he had from his year or two of study. He also looked at the timing of different flower species. So here's one of the bluebells, the glacier lilies, the larkspur, willows, all the way through the end of the season up to some of the thistles that are visited by hummingbirds, and plotted the peak flowering date for these different flower species and how long are their corollas. And you can see that there's a pattern of longer flowering corollas later in the season. He, in addition to collecting the blood samples for their genotypes and keeping track of what date he collected caught the birds, he also used a piece of sticky tape to collect pollen off of the bills of those hummingbirds, and then identified what species of flowers were they visiting. And so here's a graph that shows how much of the Ipomopsis or scarlet gilia pollen was present on birds of different length bills. And you can see that birds with longer bills tend to visit more of the Ipomopsis, whereas birds with shorter bills tended to visit, I can't quite see it. Let's see if you can see the legend there. I can't quite see the legend, but I think that was the bluebell pollen. So there's some variation in the pollen on these birds depending on how long their bills are. Turning to his genetic data, he looked at the genes that seem to be associated with both bill length and arrival time. And so in his study, here's 10 different genes that he found in the hummingbirds that seem to influence how long their bills are. Previous studies had identified a number of those same genes in influencing the bill length of other different bird species. And then in terms of arrival time and their migration dates, he found two genes that seem to affect that. And there are also some genes that have been identified as influencing migration dates of other species. because he had so many different genes that he was looking at, he was able to calculate what was the inbreeding coefficient. And it turns out that the inbreeding coefficient is quite low for this hummingbird population, which is a good thing. It means that there's not much inbreeding going on. If there were a lot of inbreeding going on, that would not be good for the genetic diversity of that hummingbird population. Another category of research that's been going on with hummingbirds at the biological lab is with regard to their vision. And let me give you a little bit of background, but explaining what I mean by non-spectral colors. So a non-spectral color is a color that is not present in their visible spectrum. So you probably all remember red, orange, yellow, green, blue, indigo, and violet as the spectral colors. But there's some colors that are not in that list and can't be produced by a single wavelength or a narrow band of wavelengths of light. So these kinds of colors that are evoked by a single wavelength of light are often described as being produced by monochromatic light, just one wavelength of light. But colors like magenta, pink, cyan, and brown are examples of non-spectral colors. So brown's not part of the rainbow, for instance, and they're produced by combining different wavelengths of light. So blue and red together produce magenta, red and purple produce pink, and so on. And it turns out that wild hummingbirds can discriminate some of these non-spectral colors. And that's research that's been headed by Cassie Stoddard, who's a faculty member from Princeton University. Hummingbirds can see 4 different wavelengths of light, including ultraviolet light, which we cannot see ourselves. We can see some of the ultraviolet reflectance patterns on flowers or hummingbirds by using photography, but we can't see those patterns with our naked eye. And so what she's done in some of her experiments is to have 2 different light sources, one light source with a control of water as a resource in front of a feeder with water, and the other light source in front of a feeder that has sugar water. And so she could use those different feeders and their different light sources to test what colors can hummingbirds see, and look at some of these different color combinations of non-spectral colors. So for instance, she might offer them red as a reward versus green with just water. And it turns out they're pretty good at discriminating that. But when you get into these non-spectral colors, they're also still pretty good at discriminating the difference between sugar water and water with those light signals. And then controls where you have nulls, they can't tell those apart, which is a prediction. So these different avenues of her research on hummingbird vision include behavior, what colors can they discriminate, foraging, what flowers and what colors are they visiting, the morphology of the hummingbird eye, how do their cones and rods work in order to see these different colors, their neurology or neurobiology, how do their retinas transmit information about these colors to the brain and what parts of the brain are involved, and then also the genetics. Are there differences among hummingbird species in their vision? So looking at all these different levels of biology with regard to hummingbird vision. A little side project that I've been working on has to do with where do male broadtails spend the night. This project was one that Bill Calder back in the 1970s came up with. He noticed that male hummingbirds, when they were leaving his feeder at dusk, seemed to fly up in altitude. So he tried hiking up the hillsides at night or at dusk to hear how high did the hummingbirds go, the male hummingbirds. He was never able to figure out an answer to that, but I think I have one answer, which is that a male hummingbird spent the night at 10,624 feet. And the reason I know that is a radio that I had put on that hummingbird fell off at that altitude. I put the hummingbird, the radio on down here at my cabin at about 9,600 feet. And then I took temperature loggers, put them down at the East River at my cabin and all the way up to where this bird spent the night and measured the temperatures. And it turns out where that male spent the night is the warmest altitude in the valley at night in July. If you go higher, it gets colder. If you go lower, it gets colder because of cold air drainage. So I think what's happening is that the males know this, and they're willing to fly 1,000 feet up the side of the valley at night to spend the night where it's warmer. The females, on the other hand, are tied to their nests at the bottom of the valley, and many of those nests are right next to the East River or some of its tributaries. And as I explained earlier, I think that may be because that then gives them easy access to the aquatic insects that they need for feeding their young. So I'm hoping to repeat that and get one more than the one data point I have, which I got several years ago, now that radios are even smaller. This is a picture of a solar-powered radio tag next to my finger. Break the radio antenna off here, can glue it onto the bird. And these are the radio tags that you might have heard about in the news last fall when they were put onto migrating monarch butterflies. And it turned out they were able to track some of these migratory individual migratory monarch butterflies for thousands of miles because they're using these radios transmitting signals. So we may be able to do that next fall with the migratory broad-tailed hummingbirds, or I may be able to do this during the summer, looking at where to find out more about where they're spending the night. So this new technology is pretty amazing. Those of you that are birders may have heard of MODIS stations. MODIS is an international project that now has a couple of thousand of radio stations, radio receiving stations around the world. But this is the only MODIS station in Delta or Gunnison counties, thanks in part to a grant that I got last year from Colorado field ornithologists. So here's the station set up as it is right now at my house in Delta County. If you look at that, maybe you can see that little bump there on the horizon. That's Uncompahgre, which is 55 miles away. So I have a good field of view here. Here is the same station set up in Gothic during the summer, and here's Crested Butte Mountain, maybe about, I think it's about 15 miles away. I set this up in the middle of the summer, and although we did detect the radio tagged hummingbirds that we tagged, I did not get any radio tagged migratory birds. I'm hoping that here where I have the station set up this spring, I'll be able to detect some radio tagged migratory individuals of other species. But then I'll move the station back up to the higher altitude to use for the hummingbirds, but also we're going to be tagging ground squirrels and potentially some other species up in Gothic this summer. Not only hummingbirds are changing their phenology of their migration, but also other species like robins. So here's some data again from Billy Barr showing that robins are now arriving weeks earlier than they used to at the biological lab. And I spent the winter of 94, 95, which is a very late spring, and opened up the window curtain in our bedroom in May. This was probably mid to late May. And here was a robin. And it was not going to find any worms here because there was still about 4.5 feet of snow on the ground. So let me give some concluding points here. Migratory hummingbirds are changing their phenology, their timing of their arrival. The wildflowers that they depend on are also changing their date of flowering, but not in synchrony with either the hummingbird migration. And so these two different kinds of phenology are now becoming mismatched in terms of their timing. The floral resources, all these different wildflowers, when they come into bloom, their phenology and their abundance depend on environmental cues. In particular, how much snow we get and when that melts, and those variables are changing. climate change is likely to produce earlier and less abundant flowering of some of these hummingbird flowers, because I've been able to show that the less snow there is, if the fewer flowers there are for things like the larkspurs. And that raises the question about how will this coevolutionary relationship between plants and pollinators change in the future. That means that biologists who are studying at the Rocky Mountain Biological Lab have lots of interesting things that they can be looking at. And these genetic studies of hummingbirds that I described are providing some new insights into both the hummingbirds and the flowers that they visit. And hummingbird vision is a current focus of the research up at the biological lab. And the banding and recapture data that we've been collecting has a potential to help monitor the ongoing changes in the hummingbird phenology, their arrival dates, also trying to make sure that their populations are remaining in good shape. We're getting a little bit of insights into Rufous hummingbirds and Calliope hummingbirds as well. So those are some of the interesting conclusions I can point to. This research is still all ongoing, and I hope I'll be able to keep collecting data myself for quite a few more years. And I'd be happy to try and answer any questions that you might have about it. If you do a Google search on my name and Rocky Mountain PBS, you can find a little documentary that Rocky Mountain PBS put up online in August of 2024. That's my older son who's continuing some of this work that is ongoing at the biological lab. And Rocky Mountain PBS also has a nice little video online about their series called Colorado Experience about the Rocky Mountain Biological Laboratory, and you should be able to find that pretty quickly too if you do a search online. So I'll stop here and answer any questions.
Thanks, David. That was a fantastic introduction to a wide range of aspects of the biology, the ecosystems up in the high mountains. Have you seen changes in nectar volumes or sugar concentrations in hummingbird visited flowers as a result of earlier flowering or years of low snow?
Great question. The answer is no. I have not collected that information. There are some researchers at the biological lab who have been collecting nectar, so we have a pretty good idea what the average is in terms of nectar production by a number of these flowers, but nobody's been doing it long term enough to know whether that's changing.
All right, and you're getting some thanks in the chat and some waves. People
I
have
think one of the names I recognized there was Sophia Todorov, who's one of the winter caretakers at Gothic, and she has helped a lot by helping count flowers for us just as the snow is melting and while the road is still closed. And
sufficient funding to keep all the research working.
another great question. I did acknowledge in my first slide the National Science Foundation. They have most National Science Foundation grants for ecologists are for about 2 years, if you're lucky, 3 years. But they do have a special program for long-term projects. And for that grant program, you have to have at least 6 years of data before you can even apply. And you have to write a 10-year research plan. And we were very fortunate to get one of those grants. And we just learned last summer that we're going to get 5 more years of funding. So at least for 5 more years, we're pretty well set. I've actually started with my wife, created an endowment at the Rocky Mountain Biological Lab to help support long-term research, including the projects I've talked about. So hopefully, at some point, we'll have an endowment there to help keep this work going if and when we lose our National Science Foundation funding. There is a visitor center in Gothic at the biological lab, which is very popular and has some displays about the work that we and other biologists are doing in Gothic. You can also sign up for tours. It sounds like maybe her group has already signed up for a wildflower tour. There's also the the Crested Butte Wildflower Festival, and the lab helps out with that. So there are a variety of ways that people can learn more about the work that we're doing in Gothic. Those of you that are particularly interested in birds, if you're getting to Gothic, get in touch with me, and maybe you'll have a chance to come observe some of the banding that is going on there.
That's been a fantastic program, and thank you all for joining us for this evening. We hope we'll see you in future CFO programs.
Thanks for hosting it.